Welcome back. This is lecture 4. You'll remember at the end of the last lecture, we were talking about the structures that increase the surface area of this inner lining of the small intestine. And we talked about the fact that within the small intestine, there are these large folds called plicae circulares. And then we have medium-sized folds along the large folds. And these medium-sized folds are called villi. And then finally, we have small folds along each of the medium folds, along each of the villi, and these small folds then are called microvilli. So we're to the point now where we're going to talk about the secretions of the small intestine, and at this point, realize that you won't need to refer to any further figures in your textbook for the rest of the chapter. So, secretions of the small intestine. Take a minute here and think back to laboratory and think back to the model we looked at of the wall of the small intestine, that model with the 3 villi sticking up on the top. You'll recall then seeing, and I'm to the handout now, to the lecture outline, and so secretions of the small intestine, you'll recall seeing on that model then that we have goblet cells in the mucosa. And then notice also now on your outline, in addition, we have other cells that secrete mucus. And these are cells called Brunner's glands. These we didn't talk about. These we didn't see on that model. These would be located in the submucosa. And then we have these structures called intestinal glands, and you may recall again seeing these on the model. The intestinal glands, also called crypts of Lieberkühn, are at the bases of the villi. And again, just following along on your outline, these intestinal glands secrete a watery fluid, so picture that. These glands, now these clusters of cells, secreting a watery fluid into the lumen, into the place where we have by now a bunch of fairly well digested nutrients and a bunch of fairly small nutrient molecules. And back to your lecture outline then, just reading along. So enzymes embedded in the epithelial lining of the mucosa secrete digestive enzymes. And these include peptidases, which, as we've talked about earlier, split peptides into their component amino acids. These epithelial lining cells, again, the cells adjacent to the lumen, also secrete lipase, which we've talked about before, functioning to split fats into fatty acids and glycerol. And something new now, these epithelial cells lining the small intestine also secrete the enzymes sucrase and maltase and lactase. And as you might guess from the names and from knowing that these are digestive enzymes, they're breaking down larger molecules into smaller molecules. So sucrase, maltase, and lactase then respectively split the disaccharides sucrose, maltose, and lactose into their component monosaccharides. So at this point, the final step of digesting carbohydrates, we're to the point of taking disaccharides and breaking them down into monosaccharides. Back to your outline then, the smallest intestine also produces enterokinase, which converts trypsinogen into trypsin. And now these nutrient molecules produced by the actions of digestive enzymes, so this combination of very small molecules that are sitting there now that have been produced within the small intestine. And these nutrient molecules then now mix with the fluid from the intestinal glands, and this fluid then, and so hence the molecules, the nutrients mixed into the fluid are reabsorbed by the villi. And we've accomplished now that very important step of getting nutrients out of the alimentary canal. And into an area where they can be picked up by the bloodstream and delivered throughout the body. Next then, regulation of small intestinal secretion. And your textbook tells you then that secretion is stimulated. And by the way, notice here now you're jotting down some notes again. So regulation of small intestinal secretion, and secretion then is stimulated by two different things. First of all, by chyme coming into the small intestine. And then also, number two, by impulses, and these are parasympathetic, so impulses, in parentheses, parasympathetic impulses, resulting from the distension, from the stretching of the intestinal wall. So just mentally picture now, here we have chyme coming into the small intestine, and just this chyme coming in contact, you don't need more notes here, I don't think, but again, picture it, this chyme now coming in contact with the wall of the small intestine causes secretion from the small intestine of these various substances we were just talking about. And then also picture the chyme coming into the small intestine, and as a result, the small intestine becoming more full and the walls stretching. And again, this stretching of the walls of the small intestine stimulates these parasympathetic impulses, which also stimulate small intestinal secretion. Looking now at absorption in the small intestine, and I think we've talked about that in as much detail as I want to. You might just want to jot down in your notes a reminder then, something we talked about previously, and that is the fact that most absorption occurs from the small intestine. Movements now of the small intestine. So number one, you'll recall mixing movements that we talked about generally early in the chapter. And so this mixing is going on in the small intestine. I'd suggest not going back at the moment, but eventually you might want to just turn back to figure 17.4 on page 657. And again, there'll be a diagram of this mixing that we talked about going on within the small intestine. Number two, then, you'll recall we also talked about the fact that propelling movements occur along the length of the alimentary canal, propelling, just pushing the food along. And specifically, in the small intestine, we have very weak, very short waves of peristalsis of these propelling movements. And this results in slow, continuous movement through the small intestine. Finally, end of the line, looking at the large intestine. And in terms of functions of the large intestine, there are three basic functions that you'll want to jot down. One is the absorption of water and electrolytes. The absorption, again, of water and electrolytes. Number two, the large intestine provides a home for bacteria and one benefit of these bacteria is the production and release of certain vitamins. And we can then absorb and we can make use of these vitamins. And finally, the third major type of thing that the large intestine does has to do with feces. And specifically then, this third function consists of the production and the storage and the release of feces. In terms of movements of the large intestine, there are continuous additional mixing movements going on. And in terms of propelling movements, in the large intestine, we have these infrequent, strong, so-called mass movements. So instead of the weak peristalsis continually going on in the small intestine, in the large intestine, there'll be from time to time a push, a very strong push of material along the large intestine. And again, these are called mass movements. And these mass movements often occur after eating. This then is the end of lecture 4 and also the end of chapter 17.